Sanchez Rivera. Francisco is going to be speaking for us today, coming from as a faculty at MIT. But despite that, he's actually a really good guy. I'm sure you all know that when you go to see talks, like this one, the main reason you come is for the food, and then you forget sort of what the person's been talking about half an hour after the talk's over. But that's actually not the case with Francisco. So he's currently a faculty at MIT, but he actually did his grad studies at MIT with Tyler Jacks, and I still remember 12 years ago, maybe it was 11, when he gave a talk at the Broad about the sort of genome editing work that he was doing as a grad student. It was fucking incredible. Sorry. It was incredible. With the, so in an in vivo CRISPR screen, right? But one where he was doing the CRISPR editing in vivo. So typically in vivo screens, you sort of do stuff in vitro and then you inject it. But he actually did the screen in vivo back 12 years ago or whatever. So when the CRISPR technology had just come out, and so he's continued in that line of really pioneering genome editing methods. And now I have the good fortune of actually we're trying to initiate a collaboration, which maybe speaks to his poor judgment. Looking at the technologies he's able to implement, he's doing, again, large-scale genome editing approaches where he's simultaneously editing individual bases while assessing in the same experiment which guide RNAs are working and which aren't, and how effective they are. And that's just with one hand, the other one's sort of juggling. So I'm excited to see the kinds of technologies he's going to describe today, and thanks so much for coming.
Thank you. Can everyone hear me well? Okay, cool. Thank you, Ramin, for that very kind and funny introduction. I think I've known Ramin for not that long, but I think knowing him after the first time, it was clear that he was a funny person and a creative person. And I've seen him now give a couple of introductions, even just one-to-one, introducing me to someone, and it's always full of humor. So I really appreciate that. And thank you for remembering that about that seminar at the Broad Cancer Program. I think it was 2014 or so. So it's been a while. I'm getting old. Thank you for the invitation. I'm very excited to present some of the work that we're doing in my lab, which I opened 2 years ago now at MIT. And pretty much what we're trying to do is use precision genome editing approaches, which I'll describe in the next couple of slides, to try to get a genetic and variant level understanding of how cancers initiate, progress, and respond to different therapies, and how throughout evolution, how those different alleles interact with each other, and how the signatures of mutations that we see in different tumor types, why are they so different? Why are some mutations seen in only some cancers, but not others? And what are the mechanisms through which those mutations arise and are actually buffered or enhanced? So long-term goals are those. But today I'll present a few basically slides, part postdoc work that sets a foundation for what we're doing in the lab, and then an unpublished story that we're very excited that we've been cooking in the lab for now a year and a half, my grad student. Okay, so I think at Dana-Farber this needs no introduction. We are living in a really new era in cancer treatment where a patient can go to the clinic, have their tumor profiled, genetically and genomically profiled, and you can get a nice signature, to some extent a biography of that tumor and the mutations that arose to drive that tumor. And that information is now being used increasingly to guide therapy and diagnostics as well. And this is basically our motivation, the fact that this is taking place. We're gathering all this information from patients all across the world. But at the end of the day, we can only do so much with sequencing data. We need to understand what each of those alleles and variants are doing, how they interact with each other, and what that means for the development and treatment of that cancer. And again, nowadays, it's becoming increasingly clear that drugs targeting specific mutant proteins are showing promise in the clinic. Matt Myerson is here. Of course, you all know the story about different drug targets and how specific mutations can influence whether specific drugs work or not. And these paradigms fall under the umbrella of precision oncology. But of course, the success of these approaches requires that we understand the function of all these variants. And the reality is that most of the mutations that we see in the clinic, not we because I'm not a physician, but the physicians in the clinic represent variants of unknown significance. And that is a big black box that we want to tackle. And if we just look at the most commonly mutated genes across all cancers, of course, known suspects here, p53, RAS, PIK3CA, and others. If you look at the different, each gene is mutated in different ways. And so some genes will show a preponderance of missense mutations, as well as nonsense, frameshifts, and other types of mutations. Other genes will traditionally show mostly missense mutations. Why is that? We can predict. Of course, decades of work have shown that RAS is an oncogene, p53 is a tumor suppressor gene, but some genes don't really just follow the general paradigm of loss of function or gain of function. And you cannot really fully assess that based on sequencing data. If we just look at p53, which is the most commonly mutated cancer gene, it's a tumor suppressor gene, it's mutated in many different ways. So here is a diagram of the structure of the protein, and you can see that there are mutations all over the protein. But there's, of course, a few mutations that are very commonly seen in cancer patients, which are commonly referred to as hotspot mutants. And there's been decades of work on showing why those are commonly seen and what the mechanisms through which those function, what is the reasoning why cancers select to have those mutations more frequently than others. But there's still a big debate about whether each mutation that is seen in genes like p53 and across the cancer gene spectrum are redundant, or whether they may have specific functions that are unleashed depending on the context, be it cell type, tissue type, cancer type, therapy, step in the evolution cascade of the cancer, and so on and so forth. So just for this one gene, and you can do it for the remaining 19,000 or so genes, the question of, are all these mutations redundant? If they're not, what are the mechanisms through which these are functioning? Are any proteins of these mutant variants required to sustain cancer phenotypes globally, thinking about cancer hallmarks here? These are important questions that could have, if answered, profound therapeutic implications. So there's been evidence over the years, and here I'm just showing 4 examples, showing that depending on the type of mutation that you have in specific genes, you can have different phenotypes. So different RAS mutant alleles in the same residue can give you different cancer phenotypes. In this case, either response or not to specific targeted therapies. In the clinic, there's evidence that different AKT alleles can influence clinical outcomes to AKT inhibitors. Complicating matters further, there are examples now shown for p53, the third promoter, and an increasing number of genes showing that you can have more than one mutation in the same coding strand, in the same mRNA strand, and we typically refer to this as composite mutants. And they can show, in some instances, in work that I did with Barry Taylor when I was a postdoc in Scott Lowe's lab at Sloan Kettering, showed that even this single, the composite mutants can have different phenotypes compared to each of the single mutants. And this has also been shown for PIK3CA as another example. And I think there are many more genes that we'll keep seeing these types of phenotypes. So this is complicating the coding cancer genome further, because you now need to think about potentially combinations of mutations within the same gene. And of course, how do we study this? We can use CRISPR. We're living in the CRISPR era where anyone can design a guide RNA and cut a gene and either inactivate it, or if you provide a template, you can do homology-directed repair. But the reality is that the nuclease is still a relatively blunt tool to do so. When you inactivate a gene, of course, it's a random process driven by non-homologous end joining. And if you provide a HDR template, you can do HDR, but it's hard to scale. So, and the important point here of using the nuclease is that the phenotypes produced by a full gene knockout are not necessarily the same as the phenotypes that are produced by expression of a defined cancer variant. And I think this is becoming increasingly more appreciated nowadays. So how can we interrogate these genes? When I was a postdoc in the Low Lab, I became interested in using base editing as soon as it came out. Base editing is a technology that was originally developed in David Liu's lab at the Broad Institute by Alexis Komor and colleagues, where you have a fusion of the Cas9 nickase, and it also works if you have fully catalytically inactive Cas9. But this fusion of Cas to azacitidine deaminase can, when combined with a guide RNA targeting a specific sequence within the genome can bring the complex to a site and trigger this C to T or D to A conversion. And this is called cytosine base editing. And this allows you to engineer these mutations in a sequence-specific manner in any gene of interest. Another flavor that exists of base editing is called adenine base editing, and this allows you to essentially do the converse edit where you're converting A's to G's and T's to C's in the complement. And now this technology evolves at breakneck speed. I think every other week we have a new tweak to the editing toolkit that increases the efficiency, precision, changes the window and the types of nucleotides that you can engineer and the types of mutations. So keep track of the literature if you can, because even to me it's my field, it's become very confusing and dizzying over the years. But suffice it to say that it works pretty well, and early on we showed that even introduction of this technology transiently into the tissues of mice somatically was sufficient to engineer mutations, specific mutations, such that you could build specific tumor models in the pancreas, in this case, by pancreas electroporation, or in the liver by either liver electroporation or hydrodynamic tailbane injection. Each of these tumors is driven by one oncogene, in this case, KRAS G12D, and a different p53 mutation. So this system allows you to then build very specific genetically defined models somatically without the need of any strain intercrossing. And this type of work was really motivated by the work that Ramin mentioned at the beginning that I did in grad school when I was in Tyler's lab, where we were using CRISPR somatically to engineer these mutations. Now we're gaining a single nucleotide level precision for building these types of models. But what I was really interested in doing is using these technologies at scale. So I was interested in interrogating many genetic variants, thousands of genetic variants at scale in a high throughput fashion. And so in collaboration with Luke Dowd, Ted, and Bianca, when I was a postdoc in the Law Lab, we developed this approach for doing high throughput base editing, where we made a series of constructs that we refer to as sensor constructs. And this is in the context of a lentiviral construct, so that's important to keep in mind. These constructs have a U6 promoter driving the expression of a guide RNA, as well as its cognate target site. That target site is what we refer to as the sensor target site. And that target site is recognized by the guide. It's essentially the same sequence, except that it has a PAM sequence. So the reason this works is that when you express the guide RNA in cells that express the editing machinery, that guide RNA will get complex with the base editor, edit the cognate target site, and by high throughput sequencing, you can amplify this whole cassette, you can then quantify the specific fraction of intended edits as well as any unintended edit and calculate those and quantify them accurately. And because this library is agnostic to the type of editor that you're using, you can then essentially do an endless permutation of base editors that you're testing against the same library. So we did that, and that's published, but the point that I wanted to highlight here is that after showing that this works, we looked at patient data to develop a set of base editing libraries that are designed to engineer the mutations that we see in patients. But keep in mind that we can only engineer certain types of mutations that are amenable to cytosine base editing in this context. So we took mutation data, combined it with different properties of the Cas proteins that we were using, as well as the enzymatic properties of the editing module, and then build two libraries, one for humans and one for the mouse genome. And these were all informed by MSK Impact clinical sequencing data, because these were built when I was a postdoc, and then we have these two libraries that we then use for a series of studies. And I just want to highlight a few examples here of what we found. So in this case, we took mouse pancreatic ductal epithelial cells that express an endogenous KRAS G12D oncogene. So they're immortalized but not transformed. They're wild type, p53 wild type. So if you put them into mice, they don't form any tumors. We then introduce this library that I refer to as the mouse base editing sensor library at low MOI, such that every cell has a unique event that you're engineering, a unique guide sensor combination that, in addition to editing in the sensor target site within the lenti, also edits the endogenous genome. And then we cultured the cells for a given amount of time and did a simple proliferation screen looking at enrichment or depletion of specific guide RNAs and therefore mutations. So anything that enriches will be referred to as a high fitness variant, and anything that depletes will be considered a low fitness variant. And then because we also have this target site that we can sequence, we can quantify the percentage of editing. And perhaps not surprisingly, based on the genotype that I mentioned for the mouse cells, most of the overrepresented guide RNAs in this screen, and therefore mutations that we're engineering. So it's important to keep in mind that we're not just knocking out the gene, we're engineering a precise mutation. Most of the overrepresented mutations were p53 mutations. And different mutations show different levels of log-fold change enrichment. The point of this graph is to show that if you take into account the log-fold change of the guide RNAs that are enriching, corrected by the percentage of editing within the sensor target site that you include in the lentiviral sensor libraries, you can get a set of mutations that you know are being increasing in the population and that you know that are undergoing precise editing because they're nicely correlated. And there are some examples that in the interest of time I don't have time to discuss in detail, but there's also some serendipitous discoveries that we made of some unintended mutations that actually turn out to be very strong mutations in the context of transformation in vivo. And then we tested each of a selected panel of mutations. Again, these are now engineered endogenously into the genome of these cells and treated or not with a p53 activating drug called NOTLIN3. And in this case, if the cells are wild type for p53, they don't proliferate when you treat with NOTLIN. They undergo arrest or death or a combination of those. But each of these mutations was sufficient to bypass the effects of treatment with NOTLIN. And these are, again, all engineered endogenously, validating the results. And this is also true when we take these cells and put them into the pancreas of mice orthotopically. Each of these mutations is sufficient to cooperate with oncogenic RAS to produce full-blown pancreatic ductal adenocarcinoma. And it's interesting to see that depending on the type of mutation, and we know we've corrected at this point for the efficiency of editing, it's interesting to note that the latency of the disease seems to vary depending on the precise p53 alteration that you're engineering in this context. Whether this is true across the board is something that we don't know yet, but it's one type of observation that motivates us to look further into different types of alleles. So just the take-home messages of this part is that we can do these large-scale screens. We can then move rapidly to interrogate these cancer-associated variants in vivo, and that at least based on this data, not all of these mutations appear to be functionally redundant. And of course, we're interested in how each of these variants is driving the disease. And for instance, we have certain mutants that seem to be inducing a higher rate of metastasis in vivo, at least using experimental models of metastasis. This is not looking at autochthonous models at this point. But if we take this mutant, for instance, C135Y within the p53 protein, and compare to every other mutant, including E271K, cells expressing this mutation endogenously seem to be more competent at generating metastasis in an experimental setting, at least in vivo. And this is preliminary, but we know that the transitional profile of the cells that express that specific mutant is different to the transitional profiles of cells expressing other types of mutants, suggesting that there is a molecular effect that may explain these differences. And this is work that's currently being pursued by Margaret Kennedy, who is a grad student in the Low Lab back at Sloan. And beyond this, I just want to quickly mention that we're collaborating with Tom Norman, Cadence Souther, and Margaret Kennedy in the Low Lab to expand this approach, which has now been done in some context, to the single-cell genomic space, where we can then use this sensor-validated guide RNAs and other types of predictions that we've generated and tested over the years to try to get a single-cell level understanding of the molecular and even chromatin accessibility phenotypes that each of these mutants is inducing in the incipient cancer cells that we introduce them in. And this is just to show that we are able to get classification of mutants depending on the transitional profiles, but this is still very preliminary, so I just wanted to show a snapshot of that. But the point is to expand this approach significantly so that we start testing different tissue types, cell types, different genetic configurations to try to understand whether different variants cooperate differently with different genetic variants and different biological contexts to produce a unique molecular signature that may explain in part why they are commonly seen in some contexts but not others, and what are the mechanisms which, by which those arise. And these are, in the future, we want to expand this to the in vivo setting. Okay, so moving to the second part of the talk, of course, this is a very complex picture of a single gene that is mutated in many different ways, and we could only model a very small fraction of the mutations that are commonly seen in patients using base editing, even if you combine cytosine base editing and adenine base editing. So we're still left with many other types of mutations in p53 and other genes that we couldn't really engineer. And really what we wanted to do moving forward is to look at variants that are observed in patients, try to engineer all of those variants, and try to do various types of iterations to look at how drugs and genotypes interact dynamically with each other, and looking at these types of interactions in different tissues and environments. And this is one of my first grad students, Sam, who said at some point, that's a tragedy. This is one of his favorite words, and I think it's like super funny when he says it. And he said, well, you can't really model all these mutations. Let me see what I can do. Maybe we could use prime editing. And so prime editing is a relatively new technology, also developed in David Liu's lab back in 2019, that involves the fusion of a nickase, Cas9, but in this case, instead of fusing it to a cytidine deaminase, it's fused to a reverse transcriptase. And it blows my mind every time I present this slide that this actually works, because what you're doing is you're bringing this complex, Cas9 with the reverse transcriptase, to a site in the genome, guided by a special guide called the prime editing guide RNA that has, in addition to the protospacer moiety, it also has a reverse transcription template, or RTT, that encodes the mutation of interest that you want to engineer into the genome, as well as a primer binding site that the RT uses after the nick to initiate a reverse transcription reaction locally at that site in the genome. And then this mutation, through some still poorly understood flap equilibration mechanism, gets incorporated into the genome stably. So because you can encode the mutation in this RTT region, you can encode a single nucleotide mutation, a dinucleotide mutation. You can also encode insertions and deletions and combinations of these. So it's a very powerful technology, and to be honest, holy grail in terms of genome editing. If no other technology came out and we're able to perfect this type of approach, I think it'll be more than enough. Why did we turn to this technology? This is the reason. If you look at the percentage of mutations that we can model with base editing as compared to prime editing can in theory cover everything that we need to model. And of course, when we go from human to mouse, the percentage decreases due to differences in synteny and sequence conservation and so on and so forth. And early on, we did a substantial amount of just testing the technology to see if it worked. And these are just some examples of mutations in p53 that we couldn't engineer with base editing, not even combining cytosine and adenine base editing together. For instance, dinucleotide mutations coming from different nucleotides, or even dinucleotide mutations that are flanking a nucleotide that you don't want to touch. These are impossible with base editing, for instance. Yet we were still able to get them at pretty much homozygosity in these cells after Nuttling selection. But even without Nuttling selection, the efficiencies were detectable and actually much better than I expected. And this is just validation that these cells are indeed bona fide p53 mutant cells, because they don't care about Nuttling whatsoever. And we've also expanded this approach. This is in the mouse genome, mouse cells, to many other mutations that, again, we couldn't even engineer with any other approach, including this mutation that there are now drugs that you can use to partially refold the mutant Y220C in humans. Okay, so how do we then deploy prime editing for doing high throughput functional genomics? This is the strategy that Sam developed. So it's a variation of our sensor approach that we use for base editing, except that now we have, in addition to the spacer and the scaffold, we also include the RTT motif, the PBS motif, as well as a TiVo pre-Q1, which is a motif at the end of a PEG RNA that has been shown by David Liu's lab and other labs to increase the stability of the RNA such that it doesn't get degraded relative to the original version without the TiVo pre-Q. So you get more stable PEG RNA per cell and higher stability. So these are encoded within the sensor cassette, and then the sensor is on this side of the construct along with native genomic sequences for each locus that are representative of the endogenous context. And this is a modular construct, so it allowed us to iterate significantly the properties of the RTT, the PBS, and everything else that we wanted to. And again, the advantage is that when expressing cells that express the prime editor, this will get complex with the editor, edit the target site, but also go into the endogenous genome and edit the endogenous genome. Whoops. Okay. In order to do this, Sam needed to develop a pipeline because the computational pipelines that existed were not amenable to design dozens of thousands of PEG RNAs that had all these different iterations within the PEG RNAs. And then the pipeline also allows you to output sensor target size. So you essentially can get a library of whatever number of hundreds of thousands of PEG RNA sensor libraries in just a couple of minutes at this point, or even seconds. And so this pipeline, which he refers to as prime editing guide generator, or PEG or PEG, I don't really know how he refers to it at this point, but PEG takes patient data, and then you can specify the information that you want the pipeline to analyze, but you essentially specify mutations, the parameters, including the pump sequence that you want to look at. So you can use the canonical NGG pump sequence or very flexible, any type of pump sequence that you're interested in. And then you can specify how much iteration you want to do on the RTT and PBS lengths and even percentage of GC and other types of properties, as well as how many internal controls you want within the library. So silent controls, synonymous edits, and things like that. And then it gives you a library of rank PEG RNA designs, as well as all the oligos and a number of visualization tools that are pretty useful when you're designing this type of approach. And if you're interested, this is the link where you can get the Python package and all the documentation. Sam, I have to say, did an extensive job annotating all the documentation, so I would definitely urge you to look at that. Okay, as a test case, we went back to p53 because, again, there is still unclear whether all these mutations are doing the same or whether there's a set of mutations that are behaving fundamentally different. But also because it serves as a nice prototype because we can select it very efficiently with Notlink, for instance, and we can screen it in vitro significantly well. So what he did is he uses input about 1,200 mutations or so in PEG, and then design a library of close to 30,000 PEG RNAs. Why did we go so big? It's because prime editing is still relatively recent days. It's a relatively new technology. We don't really fully know yet, especially back then when we were designing this, what are the different properties and features that a PEG RNA needs to have in order to be efficient and precise. So he iterated significantly on the RTT, PBS, and sequence elements of the PEG RNA. And in total, this summed up to a library composed of about 72% SNPs, insertions, close to 10%, deletions, about 11%, and then a good percentage of neutral mutations that allow us to look at the performance of the screen. This library was covering a good amount of different types of iterations of different amino acids to other amino acids. So when I say missense mutation, there was not only one missense mutation for every residue. He actually iterated significantly. But again, everything has been here informed by the patient data. So we're only looking at mutations that we see in patients in addition to the synonymous mutations and silent mutations that we use as controls. And this is just a visual representation of that. So early on, as a proof of concept, so I'm skipping a lot of steps here, but when you do library cloning, you have these intermediate plates that you count the representation of the library. So it's essentially QC. So I asked Sam to just pick 30 or so random colonies to see if the concept was actually working. So when he picked 30 or so random colonies and then purified the sensor construct, put it into cells that express prime editor, incubated them for a number of days, and isolated genomic DNA, and then sequenced to see whether the intended locus was edited. As you would imagine, and as I mentioned early on, there's a lot of pegRNAs that don't work. It's barely above 1%, but there's some that work really well. And we know that this works both endogenously as well as in the sensor target site. So this was promising. And he also developed a simple score that I won't go into any details here that is sufficient to identify the guides that work well for those that don't. So then he did a proof of concept experiment to see if these constructs were, when you read out the sensor target site, which is a synthetic target site, are recapitulating the patterns that are seen endogenously. And as you can see here, the correlation is pretty good. So we feel very confident, even from this 30 or so colony experiment that he did, that whatever pattern we see in the sensor target site will be highly representative of the pattern that we see in the endogenous site, as shown here for 2 different time points. Okay, so then we were wondering whether we could use these libraries for actual experiments. Can we interrogate this, couple thousand of variants using high throughput prime editing? So in order to do this, Sam generated A549 cells that express the PE-max, which is one of the latest generation prime editors. Everything here is PE2 for the prime editing aficionados. We're not using PE3, we're using PE2 because we prefer that it's a single component system. So these cells express PE-max, and the pegRNAs are, along with the sensor constructs, are expressed within the context of a construct that expresses a fluorescent reporter. So we could do fluorescent competition assays, for instance. And then, so he introduced this 30,000 pegRNA library into cells that express a prime editor, cultured them for about 4 days after transduction, and also selected the cells, and then treated or not with Nodlin to activate P53, and then sampled at various times throughout the screen to look at the dynamics of editing. And to my knowledge, this is probably the first study that has looked at editing dynamics at this level of number of edits over time. So as you can see here, the editing, and this is looking at all pegRNAs in the screen, the editing increases over time, but globally maxes out about 14 to 15% or so in this experiment. So again, this is just one experiment, one study, but this shows that prime editing is not perfect, but it's still very efficient, as I'll show you, for a subset of the mutations when you incorporate the right metrics to detect the mutations. Okay, so then because we have these sensor editing target sites, can we actually use them to calibrate this data? So this is here a snapshot of how the data looks. Every dot here is a single pegRNA sensor construct, or otherwise a pegRNA. So if you were to take these 30,000 pegRNAs and screen them without any idea of whether they are editing the endogenous site or not, and whether they're editing it precisely or not, you essentially get a very noisy data set. So this is here looking at the log fold change of each pegRNA compared to an early time point at day 34 plus Nodlin. And again, we're not filtering for any percentage of editing here. And which is going to be shown here. If we then systematically apply a threshold, so whether there was editing or not in the sensor target site and whether it was the intended editing or not, you can see that the data set is significantly clean, such that you're left with the events that have, in this case at least, more than 60% perfect editing within the population. So as you can see here, you can go from like a relatively noisy data set to a very clean data set by just taking into account the sensor target site. And this is a pretty funny, as you can see, very high quality Photoshop that I did of Sam, where he says the data looks less tragic now thanks to the magic of this prime editing sensor. Okay, so this is great, right? But this is all counting pegRNAs and sensor target sites. That's what we're quantifying here. Whether this has anything to do with the endogenous gene within the context of the screen, we actually didn't know based on this data. So to look at that, what we did was extracted genomic DNA from the cells that we screened, and then performed targeted sequencing of the p53 locus to see if the edits that were being positively selected by virtue of counting the pegRNAs that were being positively selected were actually being engineered. So in other words, are we actually engineering the mutations that we intend to engineer in a pooled screening setting? So to do this, we designed primer sets to amplify exon 6, 7, and exon 10, different regions of the p53 gene where we saw most of the activity, at least at the level of pegRNAs that we're enriching. And then again, applying the filter of the sensor editing percentages, you get a really nice correlation of log2 fold change. This is here endogenous editing compared to the pegRNA counts. And this is also something that increases again over time because prime editing activity we've seen here tends to increase over time. Why we actually don't fully know, and I think this is from what I hear work that will be coming out at some point in the future. But the take-home message here is that we can get pretty much 100% of the variants that we intend to engineer endogenously if we filter for sensor target site editing. So in other words, if we hadn't included these sites, we wouldn't be able to tell what's a real hit versus what's not a real hit. Okay, so this is great, but the question is, are these actually pathogenic variants, or is this just any variant that is able to kill the activity of p53 and produce a phenotype? So for this, we selected about 29 to 30 or so mutations that were spread across the length of the protein. And this includes a combination of SNPs, some missense mutations, deletions, insertions, as well as silent mutations to use as controls. And then we tested each of these edits individually in cells expressing PEMAX by transducing each of these cells with an individual pegRNA. We allowed the editing to occur for about a week, and then we mix those cells, typically at 50-50 with the parental cells, and then treated or not with NODLIN, followed by reading the percentage of cells expressing the pegRNA, and therefore the edit, by looking at the fluorescence of the cells over time. And these are two representative examples of R248Q, arguably one of the top hotspot mutations that's seen in p53 in people. When you express this mutation, the cells are easily bypassing NODLIN very robustly. But we also saw some unexpected mutations that, based on the published literature, we wouldn't have expected that they would enrich at least this significantly. And this includes the A347P mutation, which lies in the oligomerization domain of p53. As you can see here, cells that express pegRNAs designed to engineer this mutation significantly enrich over time in the presence of NODLIN, but not necessarily in the untreated version here of the experiment. So, and of course, I don't expect you to go through all of this data, but suffice it to say that our validation rate was significantly high, where if we saw if we saw a mutation enrich in the screen, and we know that the pegRNA is editing in the sensor target site, and we use that to filter, and we then validate that, the screen is essentially pre-validated in a sense. We get a pretty good correlation rate. As seen here, when we're comparing the percentage of endogenous editing, so this is now looking at cells from the competition assay where we isolate genomic DNA from those cells, amplify each of these loci, sequence those, and then correlate those results with the percentage of sensor editing that we obtain from day 16 of the screen. So we're essentially comparing data sets that are obtained completely differently, right? Sensor editing from the screen and testing whether that is predictive of the percentage that you would see in the endogenous side. And as you can see, the correlation is pretty nice. The ones that show low percentage editing endogenously also show low percentage editing in the sensor, and the ones that show high also show high. And this is true both for missense mutations as well as insertion mutations, and even some deletions as well. And I have to say, those insertions and deletions I thought would be pretty inefficient to engineer, but some of those insertions are essentially engineered close to homogeneity after just one transduction in cells. So this is great, very technical, but does this mean anything? Is there real discovery potential to use these types of libraries? Why just not screen cDNA ORF libraries, which are arguably easier, right? You have one cDNA that encodes your variant of interest. You don't have to care about the efficiency of different guide RNAs. You don't have to really sequence anything endogenously. You have one cDNA per variant. So in a sense, it's easier to conceptualize and perform in practice. So we were interested in exploring this further. And the hypothesis was that looking at the behavior of endogenous alleles would produce fundamentally different phenotypes that you might miss if you were doing just ORF overexpression. So there's been a number of studies that have been published before. And one of them was actually from Giacomelli et al. From Bill Hans' lab a number of years ago, where they did this really comprehensive study using A549 cells, which are the same cell lines that we're using in our study, where they introduced a library of cDNAs encoding essentially every other possible missense or nonsense mutant in p53, and then either introduce it into wild type or null cells and treat it or not with nutlins. So a very similar screen, except that it was done in the context of ORF overexpression or cDNA overexpression. And these are just the results, where red denotes a high C-score. So those mutants are significantly more enriched relative to the rest. And perhaps not surprisingly, there's a lot of activity around the DNA binding domain, which is where most of the cancer-associated mutations, the hotspot mutations in p53 are observed. And other regions, including the oligomerization domain, which is here labeled as 4D, were actually depleted in the context of cells having wild-type p53. So we thought that that was very interesting because we were getting different results. We were getting enrichment of cells harboring a mutation in this oligomerization domain when it was endogenously expressed. Is there something here that is of interest? So when we reanalyze the data, at least one of the time points from our data set, reanalyzed it to calculate a C-score and compare it with the cDNA data, not everything is perfectly correlated. And in fact, if it was perfectly correlated, I would be worried that there's something going on. But what was consistent was that the mutants that were lying in the oligomerization domain showed an interesting behavior, where they tended to deplete in the cDNA setting, but they tended to enrich in the prime editing setting, again, endogenous context. And if we look globally at all the data, this behavior holds for all the mutants, or most of the mutants in the oligomerization domain, where you're looking here at differential enrichment of prime editing signal versus cDNA signal. And as you can see, the oligomerization domain mutants are significantly more enriched across the board, whereas the rest of the mutants are either close to zero or slightly depleted or slightly enriched, but it's not significant relative to the as significant relative to the oligomerization domain. And if you look at this data, again, in looking at the C-score and correlate with patient occurrence, you get a picture that looks like this, where some mutants that are frequently seen in patients are being enriched with prime editing, meaning that they're being enriched in this setting when they're expressed from the endogenous locus, whereas they're exhibiting a different behavior when you overexpress them. And again, this is all data analysis. This is all building a hypothesis that maybe there's something worth exploring here. And so we decided to, before that actually, this is just showing that if we map the delta C-score of prime editing versus cDNA, the differential scores seem to be located in the oligomerization domain or in regions that are involved interaction with the other subunits of p53. Um, and if we and actually before I show this, so what did we do? We took a set of mutations that lie across the p53 gene. We took some mutations that lie in the DNA binding domain, some hotspot mutations. We took some mutations that lie in the TAB, transitional activation domain, and we took a subset of mutations that lie in the oligomerization domain. We designed the PEG RNAs individually for each of those, and then we also cloned the cDNAs corresponding to those in a backbone that's as close as possible to each other. Red fluorescent reporter, similar promoter, and both in a lenti configuration. And we introduced it into cells and then followed the percentage of cells, the fitness of those cells over time in the context of NOTLIN treatment. And importantly, we recapitulated the results of the screen, meaning that if we introduce each of those cDNAs into cells and then treat those cells with NOTLIN and follow the fate of those cells, it correlates very well with the published Z-score. So cDNA is a very consistent result and phenotype. However, if you look here to the right, while the behavior of a subset of DNA binding domain mutants is consistent with the phenotypes observed for the cDNAs, the mutants that are in the oligomerization domain show significantly different behavior where they enrich only when they're in the context of the endogenous allele. What does this mean? And there's still multiple open questions, but that's why we did the experiment. It's consistent with the fact that if you introduce a mutant ORF for a subset of mutants that are involved in oligomerization, they show a fundamentally different phenotype. In fact, the opposite phenotype than if you were looking at the endogenous allele. And I'll explain why that is in a second. So why should we care about this? It's a very interesting technical and intellectual exercise, right? But does this matter? And should we be thinking about this for human biology? So it turns out that as we were doing these studies, these two papers came out not that long ago from Carol Priebus's lab and Gigi Lozano's lab. So these were back-to-back papers published in Cancer Discovery looking at mutants that were, first of all, observed in live from many patients. So these are patients that are born with germline mutations in p53, and they are tumor prone. And the tumors often have a mutation in p53, as well as loss of heterozygosity of the wild type allele. So these mutants in the oligomerization domain that they looked at in this paper are in this A347. I think it was D, the mutation that they looked at, but it's in the A347 residue. And first of all, the fact that they are seen in people already kind of validates the fact that they're important. But interestingly, what they found in these papers is that these mutants have a very interesting mechanism of action, because when you have a mutant subunit in the oligomerization domain, those mutants can only form dimers. So you get mutant-mutant dimers that are unable to interact with wild-type p53 subunits or even insert themselves into the tetramer. So right, if we just take a step back, the most commonly accepted mechanism of mutant p53 activity is dominant negativity, whereby if you have a point mutation or a mutation in a p53 subunit, because the active p53 transition factor is a tetramer, you have a wild-type subunit, it acts as a dominant negative, or another way of typically put in textbooks is that you poison the tetramer such that you don't have transitional activity of those target genes. In this case, what's happening is that the mutant subunits can only interact with themselves. So you have mutant-mutant dimers that are floating in the cell, but you also have the wild-type tetramer if the genotype is mutant over wild-type, or wild-type plus a mutant cDNA on top. So if you express a mutant cDNA for these mutants in the context of wild-type, the mutant doesn't really even talk to or interfere with the wild-type. And if you activate p53 with NOTLIN, what's happening is that the wild-type is inducing tumor suppression, the mutant is doing its own thing, but the net effect is that you don't get any fitness advantage. And actually, the cells just deplete in the context of the screen because the wild type is leading to the depletion in a NOTLIN-dependent manner. However, when you do endogenous editing, when you either edit one allele or both alleles of the p53 gene, so if you edit one allele plus a subsequent LOH, you're essentially mutant over null. And if you hit both copies of the alleles, like we're doing with the high-efficiency peg RNAs that we're using, you're essentially a mutant over mutant genotype. What that happens is that you then have the mutant dimers that are actually interacting in the cell, but there's no wild type. So what you get is actually a production of a phenotype that wouldn't exist if the wild type was there. And once the wild type is gone, it has a certain set of neomorphic activities that were described in these two papers. So that's a long way of saying that the observations that we found, which were really serendipitously, right? We were establishing an approach to be able to do this very large-scale high-throughput genetics, interrogate specific mutations. We did it in a system where we can select for a prototypical tumor suppressive effect. We found a full set of interesting mutants. We compared to published data and identified a concept that I think applies more broadly across the proteome, meaning that depending on the type of mutation that you're introducing into a protein and depending on where that mutation, what domain within that protein that mutation is affecting, could give you completely opposite results if you're doing, say, overexpression versus endogenous editing. To put it differently, these two mutants would have never been observed in an ORF screen because they would have been actually classified as non-pathogenic, when in reality, the mechanism and the fact that they produce the oncogenic phenotypes when expressed endogenously are what is reflective of human biology, as shown by these two papers. So we're very excited about this, and we're pursuing these mutants both for p53, but a step further is actually thinking about the proteome, right? The genes, and by the way, this is just for two different mutants in the oligomerization domain. The genes that we're interested in cancer, right, if you think about it, p53 is just one example. But if you think about chromatin regulators that are part of these multi-protein complexes where the stoichiometry and the dosage is very important, what happens if all of a sudden you're tilting the balance of those subunits by increasing the dosage by overexpression when in reality maybe the endogenous edit could give you a mutant protein that is now interfering with the pre-existing complexes in the cell and producing a physiological phenotype that you may otherwise miss by altering globally the stoichiometry and just the dosage of those proteins in the cell. If you think about proteins that have disorder regions, for instance, which are getting more and more interested, those are examples of proteins where the dosage is going to be very important. And depending on where the mutation lies within those regions, you might get very different results when compared to ORF overexpression. So I think that even though we use a human cell line and a prototypical gene, p53, scanning across the length of this gene produced a really unexpected observation that now has made us think more broadly about how to deploy this approach more systematically across the cancer genome. And that's something that we're really interested in doing. So just to summarize here, physiological gene dosage, native protein stoichiometry, and protein-protein interactions are quite important. Doesn't sound very surprising, but this is exactly what we're seeing here, and I predict it's exactly what we're going to see when we start looking at other genes and proteins that are part of multi-protein complexes. There seems to be a significant functional heterogeneity, even within the p53 mutational spectrum, but I think this goes broadly beyond p53. And again, I'm very excited because I think this concept would apply much more broadly. And we are now expanding this approach systematically, and I think it was great to talk to many of you today about potential collaborations because I think the time is right to be able to deploy this more systematically. And again, everything goes back to try to understand why are patients harboring these different mutations, what do they mean, and how can we gain a nucleotide level, really a variant level understanding of cancer-associated mutations to be able to treat cancers better. So with that, I'll thank everyone in the lab, most importantly, Sam, who is really the main person driving this work, along with Alex Wiest and Kuxin Dong, who is an undergraduate business student. many collaborators and friends of mine, funding sources, and I'll take any questions. Thank you again for the invitation.